So I think we can start. Thank you all for joining us today. Hope you'll enjoy it. I'm sure I will. And Iddo will take it from here.
Thanks, Leo. So welcome, everyone. I'm just confirming that everyone can hear me, can see the screen, and can follow the cool laser. Yes? Yeah. Okay. Awesome. Okay, so thanks everyone for joining. We're going to talk today about our antibody design platform called ConvergeAB, or maybe a slightly more exotic title, How We Go from One Prompt to a Better Antibody. The idea is to give you some insight into behind the scenes, how the models work, what we can do with them. We'll show some case studies today. We'll discuss the opportunity, the value, and kind of the frontier and where this field is going. A note about questions. So basically, there's a Q&A section at the bottom of your screen. We want to try and make this as engaging as possible for a webinar that's not happening physically. You can feel free to ask questions as we go. We have a few members of the team here online who can try and answer questions live as they come up. And also, we're going to have a Q&A session at the end where we can flag relevant or interesting questions, discuss them. We also promise that any question that is asked during the webinar and we don't have time to answer, we will follow up and answer offline. Okay, let's get started. So we're going to have a few poll questions during the webinar. Here's the first one. You can all click on the bottom of your screen. There's a section for polls. It might be under the three dots for more. The question is, what is your primary professional background? This is a question to get to know the audience. Is it experimental biology, computational biology, bioinformatics, machine learning AI, or option D, a combination of all the above? I think we'll give about 10 more seconds. I see people are answering, filling in. Looks good. This is interesting. So I see a lot of experimental biology, and then it's a tie on the first place between experimental biology and a combination of the above. Cool. So actually, this is exciting for me to see that almost half of the audience marked a combination of the above. So maybe maybe this kind of prompts a quick background story. So I haven't introduced myself. My name is Iddo. I'm the Chief Scientific Officer and co-founder at Converge. Once upon a time, I started out as an experimental biologist, then quickly discovered bioinformatics, machine learning, and fell in love with all of them together. This is back in the academia. And at some point, I had a few supervisors who told me, listen, you have to choose one. You have to be really good at one of these, and you don't want to become a duck. A duck is, the metaphor is a duck can fly, a duck can walk, a duck can swim, but a duck is not exceptionally good at any of the above. So be careful not to be a duck. Choose a topic. And actually, the credit here goes to my wife, who said, you know what? Be a duck. If you like doing everything, do everything and be the best duck you can. And I listened to her because she, of course, knows everything. And boy, was she right. I ended up becoming a duck, and personally, my background is all of the above. And it's very happy to see that more than half of the audience today are ducks. So welcome and go Ducks. Okay. Briefly, let's describe the motivation for us to found this company, to start Converge. We're a bit over 2 years old at this point. Back in the day when we started, the idea was to take all the excitement and all the innovation happening in the NLP space and to try and bring that over into the molecular biology space. The reason we thought this is a good idea is the the data is in many ways very similar between molecular biology and NLP. So you can think about the data as string-like. You can think about long context dependencies within biological data. Think about genomes where potentially genes in different areas of the genome interact to give a specific phenotype, or probably more relevant for our topic today, if you think about proteins, amino acids that might be far away on the two-dimensional space can fold in three-dimensional space to form interactions. So transformers and interdependent context is very important. Something that I think people don't discuss enough is how information-rich and discrete tokens are in molecular biology. Think about nucleotides or amino acids. In many ways, they remind me of words. They're discrete, meaning that they are not continuous. If we think about pixels in images, for instance, those are continuous and very different. But amino acids are discrete. They hold a lot of information, and they resemble words in many ways. And of course, this is probably the strongest point, is that in biology, as I see from the first poll, all of you must be aware, we are always lacking label data. So in our case, we can think about proteins where we know their function, but what we're not lacking is data in general, unlabeled. We have a lot of data, a lot of sequence data out there that's just not labeled. So the idea of applying self-supervised training that comes from the world of NLP to biological data is very exciting. So really, this is the idea around which Converge was founded. And fast forward 2 years into the future, we have this amazing team of people that I feel very privileged to be working with. We've raised to date $33 million, backed by VCs such as BVP. We have a nice grant from the Gates Foundation to build foundation models. This is who we are. Today, though, we're going to talk about the science. We're going to talk about one of the focus areas of the company, ConvergeAB. I'll briefly mention that we are active in other spaces as well. We have solutions for target discovery, for gene expression, for optimizing clinical trials. But today we're going to focus on the antibody discovery piece. And now we arrive at our second poll question. We're curious to see how many of you in the audience are currently involved in antibody design. You can read the options, either active, not yet active, have been active in the past, or not from this field, but just curious, which is also fine. Okay, nice. So we'll give a few more seconds for this poll, but it seems like we have a roughly uniform distribution between all four, which is great. Okay, so the assumption would be that, as I see many of you are actively working on antibody design, that we have good basic knowledge in the audience, which is great. And those of you who are not yet in the space, it's fine too. Okay, so here's the way that we think about what AI can do, what the technology that we are building can do in this space. So how is antibody optimization carried out today? You see a lot of iteration in the wet lab. For instance, antibody campaigns often start from immunization of lab models, which is a euphemism for mice, rabbits, sometimes llamas, if you're after nanobodies. Sometimes you start from display, phage display, yeast display. These are time-consuming, multi-cycle iterations. There are many gates during this process. So, for instance, I might screen a library of antibodies. I get some results. I look at them. I scratch my head. I think about what the next iteration should be, and we keep going like this. And I think one of the so time and effort is a big pain. Another interesting feature of antibody design is that every time we screen for one trait. We don't have good experimental setups for screening for multiple traits at the same time. And this is really one of the key features of ConvergeAB. What it allows us to do, it allows us to run in a very small number of iterations. I will show you today multiple case studies where one iteration was enough. We don't need humans in the loop. It takes hours, the antibody design step, instead of months. And we can optimize for multiple parameters at the same time. This, I think, is one of the biggest advantages of moving from wet lab into the virtual space. We can screen antibodies in silico against multiple different objectives. Now, the important thing about building this type of system is that it will not need task-specific training. The way we think about this, if every time you have a campaign for antibody design, you need to first feed it with a lot of task-specific data, then in many ways you're kind of missing the point of why AI is so interesting for this field. So whatever platform it is that you build, and we'll show you today how ConvergeAB fulfills many of these items, it has to not require task-specific training. We want a generic solution that can fit many different use cases. And this brings us to discuss our antibody design platform. So here's the architecture, and we're going to go through this slowly and gradually because this is the important piece to understand. So the way ConvergeAB is built today is one workflow running. By the way, I'll first quickly say running from one end to the other takes about 8 hours. It depends a bit on some parameters, but that's the average. And there is no human intervention in between. From input to output runs completely automatic. Let's discuss the input layer. So for input, what we need, of course, is the antigen sequence. If the antigen structure is known, then that's great, but it's not mandatory. Basically, we'll soon discuss the generator, but the generator is sequence-based. The structure is only needed for some downstream filtering. So it's not mandatory, it's optional. We do, at the moment, take a lead sequence for optimization. This is an important point, and I will touch upon the idea of trying to run this whole flow with no lead sequence. We'll get to that as well. But for now, the input is a lead sequence and the antigen sequence. These two are basically the very simple, very basic prompt for the antibody generator. This, in many ways, is the heart of the system. Here, what we have an LLM architecture that was pre-trained substantially. It was pre-trained, as LLMs often are, in a stack or a pyramid, if you like. So the first level, the first layer of the pyramid, is the widest one, and it's also the most general one. Here we train on roughly 1 trillion natural protein tokens. These are not necessarily antibodies. These are just proteins. It's the same vocabulary. We use amino acids as our tokens. And that's step number 1. And step number 2, it's the next layer in this tower, in this stack. We train on roughly 7 million antibody sequences. Here too, the training is self-supervised. In the next step, we have roughly 3 million antibody antigen pairs. This is already labeled data. So this is a relatively mature step of the training. And finally, we also have a proprietary data set of roughly 10,000 fully characterized antibodies. When I say fully characterized, I mean for features such as binding and biophysical characterization and developability. An important note here is that sometimes people see the number 10,000 and think, in one screen you can get 10,000 different antibodies. But the way we think about this, if you build your data set that way, you have a lot of data, but it's all along one axis. Instead, the way we built this data set of 10,000 antibodies, and it's constantly growing, is with many, you can think about them as baby steps. So 1 plate at a time, 2, 3 plates at a time. Each time we test different hypotheses, we locate specific data points where labeling them is expected to increase the learning rate of our base model the most. We see, okay, fork number one works, so we don't bring in any new data in that direction. Instead, we bring in data where we see performance is a bit lower, where model confidence is a bit lower. And that's what we've been doing for the past 2 years. And we're still moving in that direction. The word active learning gets thrown around a lot these days. You can think about this data collection method as active learning inspired. Okay, so all this data goes into training of our generator. And basically what the generator, you can think about it as a question answer system. So the way it works, the question that prompts the system is the antigen, and the answer that it gives is the antibody. Now, we run this prompt not once, but 30,000 times. And here too, I want to stop and say a few words about this number. So I've heard about different approaches here, and I actually think that this number here is the KPI of how fast this field is moving. If you think about the way you interact today with LLMs in natural language, you can think about Claude or ChatGPT. Basically, you prompt the system with your question, and then the system generates a single answer for you. Why does it generate only one answer? Because the technology is relatively mature, and we know hallucination rate is relatively low, so one answer is good enough. I think that's where we should be shooting. I will talk about like my projections into the future of antibody design later, but we're still not there. We're still in an earlier step where we know that LLMs trained on antibodies do still hallucinate, which is why we generate more than just one candidate. In our case, we generate 30,000 candidates that are then filtered on some downstream predictors. These downstream predictors are more explicit, more supervised models that test for features like pairing of heavy and light, solubility, thermal stability, aggregation, and so on and so forth. After all these filters, about 1,000 sequences go into our docking protocol. This is where we need the structure. Structure prediction tools today for single entities are pretty good, which is why the structure is not mandatory. Eventually, after this step, we aggregate everything that all of the characterizations for all of the generated antibodies and select the final 24 candidates for experimental testing. This runs, again, automatically in about 8 hours, and this is the system that we built. Now, the big question is, okay, this is nice, but does it actually work? So we're now at the point where we tested this flow multiple times and quite rigorously. I won't show you the computational benchmarks. I will go straight to experimental validation of this pipeline. So the first case study, when we had our first version of this pipeline built end to end, we kind of looked around the field in search for benchmarks. Many of you are probably aware that there aren't very good benchmarks yet, and maybe this is like a nudge for people in the community who are online now. I think a project of creating benchmarks would be highly appreciated. The closest thing we found was there was a competition run by a company called Adaptive about a year ago, and there was the task there was to design an antibody against EGFR that would beat cetuximab, which is a clinical antibody that sells for about $1 billion a year, to try and beat it in binding affinity to its target EGFR. There was a winner for that competition from a company called Cradle, and we thought that was an interesting benchmark. The sequences were shared. Both Cradle and Adaptive put all the information out there, so it was, and kudos to them. I think more people in our community need to be open about the sequences that their AI designs. We try to adhere to that principle. It's important that we let our peers review what we do. So again, kudos here. We took the sequence that won the competition, and we took cetuximab itself as the baseline. And if I can flip one slide back, we basically prompted the system with cetuximab itself as the seed sequence. And the antigen we gave was EGFR. We just ran this. It took 8 hours. It gave us 24 candidates, and we tested them in the lab. What you see here is the best candidate that we found out of those 24. And here are the results. So you can look at the binding affinity, which was essentially the goal here. Cetuximab binds at about 670 picomolars. Cradle's antibody also binded nice, but with a higher KD compared to cetuximab. And you can see here that the antibody we designed binded about 2 times stronger than cetuximab. Now, we repeated this a few times. We wanted to be sure because the deltas here aren't huge. These aren't orders of magnitude. So what you see here are the raw SPR curves. We have here 6 different concentrations for each of the antibodies run on the same day and the same instrument, side by side, same batch, same manufacturing. And this makes the result more compelling, in my opinion. You can see that in all concentrations, there's a very clear separation based on the K off value. So this was a result we were happy to see. But it's just one result. And maybe for some reason, EGFR or cetuximab were a bit closer to the training distribution that we've seen during training. So we immediately moved to 4 other targets, and we did the exact same there. The next targets we chose were PD-L1, spike coming from COVID, HER2, CD19. So these represent many of them are well studied, but different targets. And each one we started with a different seed antibody. You can see some of them bind very strongly, sub-nanomolar. Some had a higher starting point, like a double-digit nanomolar. And the experiment was relatively simple. For each one of them, we prompted the system with no customization, with no added training, just that same system. That's a key design principle. We prompted it with the sequence of the target. So we had these 4 targets and the sequence of the parent antibody. We got 24 candidates, but in this experiment, for different logistic reasons of what fits in a plate, we tested 10 antibodies from each. And for one, we had room for 2 more, so it's 12. You can add these, you get 42. As you all know, everything in biology works in multiplications of 48. We had 6 controls, so that's how we get to 48 here. And what's important is that we show here all the data. So this data is not cherry-picked in any way. And let's go over this plot because there's a lot of information here. So on the y-axis, we have the binding affinity, and I remind everyone that lower is better. The dashed horizontal line is the KD of the seed sequence. And then all the dots you can see on the bar are the KD of the variants that we tested. I'll start with the gray X's here on top. These represent cases where we lost binding. These are hallucinations of the model. Our model thought these would be good binders, and actually they did not bind at all. And we like to be very open about it, that this does happen sometimes. We're still at the point where we're not at 100% success rate yet. I do think we're moving in that direction, but I'll touch upon that later. What's interesting to see are all the dots below the dashed line. These are variants. These are sequence variants of the parental antibody that were actually improved. And you can see in each figure, in orange or red, I'm colorblind, sorry, you can see the best binder. So for PD-L1, for instance, you can see we have a 500-fold improvement in KD. This was the biggest improvement. And you could have maybe guessed that when we start with a double-digit nanomolar binder, there's more headspace for improvement. But what was interesting to see that even when we started with this very tight binder, so indeed some of our variants binded at similar or worse KD, but we were still able to find an almost 3x tighter binder. And you see the success rate in the different for the different antigens. So this was very encouraging because now we're not just looking at we see it working not just in one target or in one seed. We expanded 4 more targets, 4 more seeds, and this was cool. And at this point, we started asking questions, including, okay, what else can we do with this system? And a very interesting use case that came to mind was a question about humanization. Basically, can we take a mouse antibody and improve its binding while also humanizing it? So we have here 2 different objectives. Okay, one is the binding affinity, the other is the humanization. And the way we do this is that we basically allow the model to make multiple edits in the framework regions as well. And I can show you here an interesting case study where we took FMC 63. This is, some of you might know, this is a very well-known antibody used in CAR T cell therapy. It's an anti-CD19 antibody, and the one that is actually used today in the clinic is a mouse antibody. So we allowed our entire flow, the ConvergeAB platform, to introduce edits here. One objective was to raise the percent humanness of the frameworks, and you can see that we succeeded. We increased the percent humanness from 80s to high 90s. So that was a success. And at the same time, we also improved KD. So you can see here a first glimpse into this multi-parameter optimization that we were shooting for. Now, another really important feature for antibodies is their developability profile, or what we sometimes call their biophysical characterization. So we know that having a good binder is not enough. We want it to be stable, to have a high melting temperature. We want it to be soluble. We don't want it to aggregate or to interact with itself. So at this point, we started testing all these parameters as well, as our platform optimizes for all these features together. And you can see in this case, we have for hydrophobic interaction, higher is better. So we have a win here. We have a win. There was slight aggregation here for the parental antibody that was improved. The melting temperature in this case was not improved. But if we flip back to the previous experiment I showed you, we can now look at the developability parameters here. So again, we're recording melting temperature. And for the advanced users, we're also recording T onset and the temperature at which the antibody starts aggregating, and the percent monomer, which reports an aggregation, and solubility. I know this is a busy slide, but you can see that for almost all the antibodies that we generate, the green background shows similar or superior developability, meaning that while we're significantly improving binding affinity, we are also improving or maintaining the developability profile. And this is exactly the multi-parameter optimization that we've been shooting for. Okay, so now switching gears from these case studies to some thought about how we can use this system. And here comes the third, and I think maybe the last poll for today. The question is, for those of you who are actively working on antibody design, when you start a campaign, what's your typical starting point? Do you usually have an existing lead antibody? Maybe you don't, but it's easy to find. Maybe you don't have one, but if someone were able to point you towards a starting point, you'd be happy to do that, or you usually start from scratch. So we'll give a few more seconds. Um, I see some interesting. This is actually something that I'm very curious about. We see all kinds of different cases when we work with our collaborators. So I see a split at the moment. By the way, I'm not sure if the audience sees the results of the poll, so I might be narrating it for no reason. But there seems to be like the leading seem to be A and D. So about a third typically start from scratch, about a third typically start from an existing antibody, and I see about 20% that mark number C, that they don't usually have a lead, but in theory would be happy to have one. Cool. Okay. So an interesting thought. This is like a quick sneak peek to behind the scenes or under the hood of the ConvergeAB platform. So we have we have behind the scenes a very large amount of antibodies. Many of them we've curated from open source. And here it's important to say, curated from open source doesn't just mean scraping them in. In many cases, we will then also go and QA the resource from which we've scraped the antibody. So let's say we found a new data set. Something that we often do is go and experimentally test how reliable the data in that new data set is. So for instance, we would randomly sample a large data set with many antibody sequences and test them in the lab and see how much we can count on how reliable that data set is. Some of these antibodies come from our proprietary collection. And in total, we have about 1.3 million different antibodies that are confirmed to bind about 4,500 distinct antigens. The vast majority of these are human antigens. And here you can see the number of antibodies per antigen. So many are, of course, singletons where we only have one seed sequence per antigen, but you can see that for about 1,000 antigens, these are the antigens that are more interesting and more well studied in the community of drug developers. We have above 10 and in many cases above 500 different seed sequences that one could use as a campaign. The reason I think this is interesting to look at is that sometimes we work with people and they say, well, actually, I don't have a lead. And in those cases, sourcing a lead from this database is something that we support. And in many cases, it can shorten timelines of development. And it's an interesting feature to think about. Okay, so now switching gears a bit, I promised I would talk a bit about the idea of basically starting with no lead. So this is what people sometimes call de novo antibody design. However, the word de novo design gets thrown around a lot these days, and I wanted to start with a definition. So the way we think about true de novo design means I don't have any starting point, and I also don't have any task-specific data. If I have a lead sequence, or if I have a lot of data that is specific to the task, and I try to find another data point within that data set, that's not what we mean when we talk about de novo. De novo means I really have a generic model that can be prompted only with the antigen and with no antibody seed. So this is an interesting direction. I can share openly that we are working on this heavily, and we have some very interesting initial signs. I know that many people today, it's a hot topic, and many people, many labs, many companies are talking about de novo design. I do feel comfortable sharing here with the audience some thoughts about this. So let's start for a moment with a quick journal club. These are stats from the Nature paper from the Baker lab that came out about 6 months ago, from the RF antibody paper, where the Baker lab in University of Washington attempted de novo design. These results are peer-reviewed and I think are highly reliable. Here's the data. So you can see different targets that they went at, that they tried to design truly de novo binders for. Here you see the amount of antibodies that they screened. You see the screening method. So in some cases it was like real SPR, and in other cases it was a display. And here are the number of confirmed binders that were found. So you can see, and importantly, all this is for VHH, okay? For a nanobody where all we have to design are 3 CDRs. You can see the success rate is between 0 and 2%. In many cases, they get like 1 out of 9,000, 5 out of 9,000. These are the reported success rates for VHH. They also, in the same paper, attempt to build 6 CDRs de novo in a single chain FV. And they report openly that there are no hits. This is the most recent peer-reviewed publication on de novo design. Now, some other tools out there today make different claims about being able to do de novo design. In most cases, the sequences are not shared. Only the success is shared without the sequence itself, without the code. I personally find many of these reports very hard to replicate. Wherever some code is shared, I can share that we at Converge have attempted to recreate some of these results, and so far have not been successful. So this is kind of an honest, the way we honestly feel about the status of de novo design. It is a focus in our R&D. We are actively working on it with some interesting initial results. By the way, if anyone on this call is interested in collaborating in a design partner manner around de novo design, we're very happy for those types of collaboration, and we're actively running multiple of them. Okay, so this is this. The last slide I'll show before our Q&A session is a slide talking about a few thoughts regarding where the market is going. So the way we like thinking about this, I think in many ways we're still very much in the GPT-1 or pre-GPT-1 era of antibody AI. If those of you who are good at quick math can see that actually our antibody-specific data in our data stack culminates to about 1 billion tokens, which was the size. 1 billion tokens was roughly the size of the training data set for GPT-1, which we all know hallucinated quite badly. It was interesting, but had a lot of hallucinations. In many ways, data still remains limited, and we think that like we and others are working on expanding the data sets. But another really important thing that is happening, and we at Converge are very much focused on it, is developing modern pre and post training techniques that fit biology much better. I think a lot of what people in the field are doing today is taking architectures that we know work well for natural language and just shoving a lot of molecular data into them. It's helpful. It's a good starting point. I think many of us are aware that it doesn't just work out of the box. There are subtleties about biological data that are different in their context, in their structure, in their grammar from natural language, and we're still very much in the infancy of understanding what techniques are best. But I expect both the data wall and the training techniques to move very fast and for the models to improve probably much faster than many of us think. The KPI for this is very interesting, I think, and many people don't think about it this way. The KPI for this is how many variants we actually generate. I'll remind you, we typically generate 30,000 variants in a typical run. I sometimes hear people bragging about their system generating 5 million candidates, and then they screen those. And that's, when I stop and think about it, that's actually not a good thing, right? Like generating variants is easy, right? The reason we need an LLM is we can think about design as a search problem. So basically, I can algorithmically generate all the different combinations of amino acids in the world and screen that. That, of course, won't compile. But the more the LLM can reduce the search space into, first 5 million, then 30,000, maybe down to 100, and eventually we'll get to the point where NLP is today, and we'll be able to only generate one sequence, and we know that one sequence is going to be a winner. I think that's actually the number we should be monitoring to understand how quickly the pace is picking up in this field. And I can share here that we've downsized that number. And up until a few months ago, we would generate 100,000. Now we're down to 30,000 because our confidence in the model keeps growing. So models are going to quickly improve. But at the point where we are now, they still hallucinate. The good news, I think, is that maybe unlike in NLP, even if our hallucination rate is high, let's say, 90% hallucination for NLP would be extremely high. Imagine like 90% of your conversation with ChatGPT would be nonsense. You wouldn't use the product. The good news about our field is even if I have 90% hallucination, it means that from the ConvergeAB pipeline, if I give a collaborator of mine 24 antibodies to test, 2 or 3 are actually going to work and give them exactly the antibody they need with the binding and developability and biophysical characterization profile that they were looking for. And this actually is quite awesome. I've showed you today some case studies where the hit rate was much higher than that, 30%, 50%. So the good news, while the field is moving rapidly and has a lot of headspace for improvement, the current state of technology is already good enough to bring very valuable technology into products so everyone can enjoy and start using. With this, I think I'll pause and we can have a Q&A session. I'm happy to take questions. I'll address the teammates here. Leo, if you want to drop some questions for me in the box, let me know how would be best to go about this. Okay, so in the meanwhile, I think I found the Q&A box myself, and I will scroll through it. So I see many of the questions here were already answered by members of the team.
I think we can answer a few questions to the entire forum in case they haven't visited the Q&A section.
Okay. So there's a question here. Maybe I'm reading from the end. How are you enabling patents? Okay, so first of all, I'm going to try and make sure I understand the question. So I assume the question is like, how do we create freedom to operate on the generated sequences? If we start from a lead sequence and that lead sequence happens to be patented, then how do we create freedom to operate FTO on the generated sequence? That's a very good question. The answer is that the seed really just is used as a starting point for the generation method. You can see and you can welcome to visit our website. All the sequences that we generate, we also shared, and you can find them there. The edit distances from the seed sequence are quite dramatic. Some from as low as 6, 7, 8, 10 edits, mostly in CDR regions, up to sometimes 20, 30, or in the humanization case study I showed today, we had I think 45 edits. So the answer is that we diverge quite significantly from that starting point. We still think that that starting point is helpful in steering the generation in a general trajectory, but the final sequence that you actually receive is quite diverged from that initial starting point. Okay, I see here another question. Can you use your platform to generate nanobodies? And have we tested this in the lab? Great question. The answer is yes. We do support nanobodies. I see Omer just answered. Yes, we support nanobodies. We have tested this in the lab. We've seen some good results for VHHs. So yeah, simple answer is yes, and we're happy to also chat offline and dive deeper into that. There's another question here. Do you succeed in all cases, or are there cases that are too hard? So I like that question. And yes, indeed, we do not always succeed with everything. I think also today we showed a few cases where antibodies that we generated did not bind. There's a question about the definition of success. If we generate an antibody that doesn't bind, do we think about that as a failure, or as long as there's at least one satisfactory antibody in a campaign, then we think about it as a successful campaign? But the honest answer is that yes, this is very early. The technology is early. We're now running this on multiple use cases and with our collaborators internally, and we do not always succeed. We do have we are becoming very confident in our success rate, and you can see this in the way that we run our internal case studies. We're generating less and less candidates. We're testing in the lab less and less candidates. We're not testing 96. We're testing 10 every time. So the confidence in the technology is growing, but yes, still there are some cases where we did not succeed. Okay, there's a question here. Which step is more challenging or costly, generating the candidates or predicting or running the predictions? This is also a good question. So in fact, there's not one very heavy step. The LLMs, as many of you might know, the LLMs are heavy to train. So training them can take up a lot of compute and have significant cost, as we all know. But then running them in production, what we call inference, is not necessarily extremely heavy. So I wouldn't say that the generation is extremely costly. It does take some time because we are generating a fairly large amount of sequences still. Filtering takes time, especially we have a docking step, which also runs some orthogonal biophysical models, which tend to be computationally heavy. So I'd say there's not one major bottleneck in this process, but all of it takes some time. Okay, I'm searching for some other questions. Okay, I see almost everything is answered. Do we own our own wet lab? The answer is no. We collaborate with many different wet labs in many different places. Okay, maybe I'll open to Leo or Dov, one of the panelists, if you want to point me at specific questions. I'm afraid I'm missing maybe some questions here.
There's a question around, have you played with other formats like drug conjugates?
Yeah, good question. So we haven't, so we are currently not like designing specifically the ADC, but we are, the ADC, the antibody component within the ADC is, of course, relevant for this platform, but we're not doing at the moment any design of the linkers themselves, and we're happy to take offline if there are any more specific questions about this.
Another question is, what percentage is docking based?
Docking, maybe the way we think about docking, we think about it as yet another filter. So again, we generate antibodies, then we start filtering them with many different, more explicit models where kind of inductive bias is put into the process to try and minimize model hallucination. One of these filters is a docking filter. And then eventually the final candidates are scored by all these different filters. Docking gets a similar weight to the weight of many other filters. And it's just one of the judges. So I hope this answers the question. So I think with this, I will move to the last slide. We'll say thank you for everyone for joining. Hopefully, this was interesting and engaging. We're very happy to keep the conversation going. Please feel free to reach out on any of these means of communication. And the recording of this session is going to land in your inbox shortly. Thank you very much. Leo?
Thank you all.